I think the diagnostic landscape of psychiatry often feels, well, it's a stark contrast to the rest of medicine, isn't
Yeah,
it?
absolutely. Massively different.
Right, because if you're, say, on an orthopedic ward and a patient comes in with a fractured tibia, the process is, I mean, it's almost entirely mechanical.
Yeah, you take an X-ray and it's right there.
Exactly. You locate the structural failure, you point to this jagged white line on a screen, and it's just broken or not broken, it's binary. And there's this undeniable comfort in that kind of visible truth.
There is, yeah. We inherently gravitate toward pathology that we can sort of hold in our hands or, view under a microscope.
It gives an illusion of certainty.
Spot on. The illusion of absolute certainty. A blocked artery, a ruptured appendix, a high viral load. But when we transition into psychiatry, into neurodevelopment, major mental illness, human consciousness, that mechanistic framework just completely collapses.
Because the broken part isn't there.
Right. The broken part simply isn't there to be found.
Which forces us to really reckon with what psychiatry actually is. If we're going to dive into this, we have to start by explicitly framing the discipline for you. So, psychiatry is the study of how the brain, the mind, and lived experience interact, and critically, how that incredibly delicate, dynamic process can go wrong.
Yeah, we are dealing with moving parts.
Moving parts, exactly.
We're dealing with systems.
Yeah.
And to understand those systems, we need to just completely discard those old mechanical metaphors. The human brain is not, it's not a car engine.
Right.
It doesn't have isolated modular parts where a single stripped gear brings the whole vehicle to a halt. If we want a useful model for this deep dive, we have to view the brain as an orchestra.
I love the orchestra metaphor. I think it's brilliant because it introduces the concept of time.
Yes. Time is everything here.
Right, because in a symphony, you can have this masterful violinist playing a flawless melody. You can have a cellist executing their sheet music perfectly. But if they're isolated, or more importantly, if they're not playing in time at the right volume and in the sequence the conductor demands,
then the perfection of the individual instrument is totally irrelevant.
it's just noise.
Exactly. The harmony is entirely dependent on coordination. And a psychiatric disorder, when you view it through the lens of modern systems neuroscience, is very rarely a broken violin.
The cells are fine.
The individual cells, the localized brain regions, they're often physically intact. They're functioning exactly as designed. The pathology lies in a loss of temporal harmony.
So it's like the string section rushing the tempo while the brass section is playing a completely different movement.
Spot on. The music descends into dissonance, not because the instruments are flawed, but because the relationships between them have fractured.
So we're moving from parts to patterns, and that is the central mission of this deep dive today. We are exploring the Kaplan and Sadok chapter on basic systems neuroscience.
Which is such a foundational text.
It really is. We're moving beyond the isolated neuron to understand how distributed large-scale circuits actually give rise to human thought, emotion, and behavior.
And to do this, the text introduces this great conceptual framework called the circle of being. It's a way of visualizing the distinct levels of biological organization.
Right, and how they all feed into each other.
Exactly. How each level mechanistically influences the ones right next to it, creating this continuous feedback loop.
Let's trace that loop, actually, because it highlights a massive blind spot in our current science. So at the foundational microscopic level, we have genetics.
Right. DNA and RNA variations.
Which influence the structural properties of cell membranes, ion channels, neurotransmitters, and those molecular components dictate how individual synapses function.
Yeah. And the synaptic function dictates the firing properties of a single neuron. Does it fire easily? Is it resistant?
And those single neurons wire together into local microcircuits, which then connect to form vast macroscopic brain networks.
And then we make the leap into the macroscopic world. The activity of those large-scale networks drives individual human behavior.
But the loop doesn't stop there, does it?
No, it loops back. Your individual behavior dictates how you navigate social interactions. And crucially, those social interactions feed back down the chain.
So social experience alters behavior, which changes network activity.
Which can actually induce synaptic plasticity and physically alter gene expression over time through epigenetics.
The system is constantly modifying itself.
It is, but mapping this circle of being reveals the profound frustration at the heart of modern biological psychiatry.
The missing middle.
The missing middle.
Huh.
We possess astonishing technological tools to examine the extreme ends of this hierarchy, but we're essentially blind to the middle.
Let's talk about that, because it explains so much of why translating research into clinical practice takes decades. On the microscopic end, we've achieved miracles, really. We can sequence entire genomes in hours.
Yeah, we can map the specific binding affinity of a dopamine molecule to a D2 receptor down to the angstrom. We know the exact cascade of intracellular proteins that activate when that receptor is stimulated.
And on the macroscopic end, we're equally well equipped. We can put a patient in an fMRI scanner and watch broad swaths of the prefrontal cortex consuming oxygen.
Or use an EEG to record massive, slow electrical waves sweeping across the scalp.
Not to mention the ultimate macroscopic tool, the clinical psychiatric interview.
Exactly. Where we observe the complex behavioral output of the entire system.
But knowing the molecular structure of the violin string and then recording the muffled sound of the concert hall from outside the building, it doesn't tell us how the musicians are actually playing the symphony.
No, we have a vast, technologically impenetrable gulf between the receptor and the behavior.
And that missing middle is defined biologically as the temporally organized action potential discharge within ensembles.
Right. We're talking about how specific subpopulations of single neurons, often tens of thousands of them, fire in highly coordinated millisecond by millisecond patterns.
Which is the thought itself.
It is the thought. It is the memory. It is the delusion. Without understanding how that ensemble timing works, trying to connect a microscopic genetic mutation directly to a macroscopic symptom like paranoia is, well, it's an exercise in immense speculation.
If I can use a slightly different analogy to ground this for you, it feels like trying to understand the daily functioning of a massive, congested city.
Ooh, I like this one.
Right. So we have molecular biologists studying the chemical composition of the asphalt on the roads, and we have macro-level researchers looking at blurry satellite imagery showing heat maps of traffic congestion.
But the missing middle means we cannot see a single individual car.
Exactly. We can't see the traffic lights changing, we don't know who is merging into which lane, and we definitely can't see the near-miss accidents at the intersections.
That captures the dilemma perfectly. Systems neuroscience is the discipline built to finally place cameras at those intersections.
But we can't exactly implant arrays of thousands of microelectrodes deep into the cortex of a healthy human volunteer, can we?
Unsurprisingly, no. Ethics boards tend to frown on that. So we are forced to rely heavily on animal models to bridge the gap.
Which brings up a massive conceptual hurdle. The sources spend a lot of time deconstructing how we historically use animal models in psychiatry. The whole idea of the schizophrenic mouse or the depressed rat is just thoroughly dismantled in this chapter.
It has to be dismantled because the premise is conceptually flawed from the ground up.
How so?
Well, complex psychiatric phenomena like the experience of highly structured persecutory auditory hallucinations or the profound existential anhedonia of major depression, these are uniquely human experiences. They're intimately tied to human linguistic and cognitive architecture.
Right, a mouse doesn't have language.
Exactly. We have absolutely no empirical basis to assume a rodent possesses the mental capacities required to experience these states.
And even if a mouse were experiencing a paranoid delusion, how would it possibly tell us? It certainly wouldn't manifest in a way that aligns with the DSM, the Diagnostic and Statistical Manual. You can't use a checklist of subjective human experiences to diagnose a rat.
You can't. And this specific failure is driving a major philosophical shift in the field, championed by initiatives like the NIMH's RDOC, the research domain criteria.
Right.
The argument is that if we define a disease purely by subjective, culturally influenced symptom clusters in humans, it becomes impossible to model it in an animal. We have to shift from subjective phenomena to objective, measurable biological criteria.
So, if the mouse isn't actually experiencing schizophrenia, what is the point of mutating a mouse's genome with human risk variants? The chapter uses this specific term, reagent. We need to view the animal not as a patient, but as a biological reagent.
Yeah. In chemistry, a reagent is a substance added to a system to cause a chemical reaction or to see if a reaction occurs.
So in translational systems neuroscience, the animal is a living reagent.
Exactly. We're using it as a biological tool to test highly specific mechanistic hypotheses about neural circuits. We do not care if the animal is psychotic.
We care about the circuit.
We care about how a specific genetic deletion or a developmental insult alters the timing, the excitability, and the synchrony of its hippocampal prefrontal networks.
But even using animals purely as circuit-level reagents comes with formidable genetic hurdles. The text highlights two major confounding factors. Pleiotropy and penetrance. Let's break those down.
Sure. So pleiotropy is the phenomenon where a single genetic locus influences multiple, seemingly unrelated phenotypic traits. A gene is rarely dedicated to just a single function.
Right.
Furthermore, the genetic background matters immensely. A specific gene variant that influences, say, synaptic pruning in the context of a human genome might confer a completely different or utterly silent phenotype when expressed against the background of a mouse genome.
Because the systems are vastly different.
Vastly. And penetrance makes it even murkier. We know that the genetic variants associated with major psychiatric illnesses, even significant ones like the 22q11.2 microdeletion, they often have incomplete penetrance.
Meaning a patient can carry the mutation and never develop schizophrenia.
Precisely. So if you engineer a mouse to carry a low-penetrance human risk variant, the overwhelming probability is that the mouse's circuits will just develop completely normally.
Compounding all of this is the issue of genetic overlap, right? GWS studies, genome-wide association studies, have definitively shown that the genetic risk architecture for autism, ADHD, bipolar disorder, major depression, and schizophrenia, they share immense overlap.
There is no single schizophrenia gene.
So if you knock out a shared risk gene like DIC1 in a mouse, which clinical disorder are you actually modeling?
You aren't modeling the disorder at all. You're just modeling a non-specific vulnerability in the circuit.
Which brings us to a massive practical challenge. If the goal is to study how these vulnerable circuits fail under pressure, how do you put a rat under cognitive pressure? I mean, I can give a human patient a cognitive control test. I can use the Stroop test.
Ah, the classic Stroop test.
Right, where the word blue is printed in red ink, and the patient has to consciously suppress their automatic instinct to read the word and instead name the color of the ink. It requires intense cognitive control and conflict resolution. How on earth do you administer a Stroop test to a rat?
You can't. You have to exploit the cognitive domains the animal naturally relies on, primarily spatial navigation and threat avoidance, and engineer a scenario that forces a cognitive conflict.
And the classic example detailed in the literature is the active place avoidance task. This experiment is fascinating. Walk us through the physical setup of this task, because the brilliance is really in the environmental design.
Okay, so picture this. The animal is placed on a large circular arena that is slowly, continuously rotating.
Like a turntable.
Exactly. And somewhere on that arena is a stationary, invisible sector where the rat will receive a mild, aversive foot shock.
So the shock zone is tied to the absolute coordinates of the room, not a specific spot on the physical floor of the moving arena.
Correct. Now, rats heavily utilize olfactory cues to navigate. They leave scent trails. On a normal stationary floor, a rat would simply smell its own previous path, realize, oh, I was safe here a minute ago, and just stay put.
But because the arena is rotating, the olfactory cues on the floor are constantly moving in relation to the stationary shock zone in the room.
The olfactory data becomes actively misleading. If the rat follows the scent on the moving floor, it will inevitably be carried directly into the shock zone.
So to survive the task, the rat has to undergo immense cognitive effort.
It does. It must actively suppress and ignore the highly salient proximal olfactory information from the floor, look out into the broader environment, and selectively attend to the distant stationary visual cues on the walls of the room to calculate its trajectory.
It has to ignore the word blue and read the red ink.
That's exactly it. It is resolving conflicting streams of sensory information by suppressing the dominant automatic response in favor of a higher-order, goal-directed calculation.
And that is the exact overarching neural process we define as cognitive control in humans.
Right. And the true power of this Regent model is that while the rat is performing this complex suppression, we can have neuropixel arrays implanted in its brain. We can record the millisecond by millisecond action potentials of thousands of neurons simultaneously.
We can literally watch the ensemble code of cognitive control as it happens.
We can, which is the perfect entry point to dive into how these ensemble codes actually function physically. How does a thought or a memory physically exist within the electrical firing of a biological system?
Right, because for decades, the dominant theoretical model was the dedicated coding hypothesis, often colloquially referred to as the cardinal cell theory.
This is the intuitive, almost Aristotelian idea that one neuron equals one specific concept.
And this concept gained huge traction in the public consciousness with the discovery of the so-called Jennifer Aniston cells. I remember reading about this. Researchers had epilepsy patients who required invasive intracranial electrodes for surgical mapping.
Yeah.
They showed these patients hundreds of photographs, and they found single, specific neurons in the medial temporal lobe that would fire action potentials only when the patient was shown a picture of Jennifer Aniston. Not her co-stars, not other celebrities, just her.
It's a remarkable finding, and we see parallels in the rodent literature, too. The discovery of place cells in the rodent hippocampus actually won a Nobel Prize.
Oh, wow.
Yeah. A specific place cell will remain completely silent until the rat walks into one specific 10-centimeter corner of a box, at which point it just fires furiously. We found head direction cells that fire only when the animal faces precisely magnetic north, and border cells that map the physical perimeter of an environment.
So the evidence for dedicated coding seems overwhelming. You have a neuron for every specific feature of the world, but mathematically that model completely falls apart, doesn't it?
It collapses under the weight of capacity limits. The human brain contains roughly 86 billion neurons.
Which sounds like a lot.
It sounds vast, but the universe of conceivable thoughts, faces, memories, abstract concepts, and specific sensory details is practically infinite. If we required a dedicated, unique cell for every distinct concept we ever encountered, we would have run out of cortical real estate in early childhood.
We'd have to overkit the neuron that remembers our mother's face just to learn a new telephone number.
Exactly. This capacity constraint necessitated a paradigm shift to the ensemble coding hypothesis.
Okay.
Information is not represented by the firing of a single, highly specialized cell. It is represented by the specific, coordinated pattern of activity across a vast, distributed network of neurons.
The textbook uses a brilliant, really accessible metaphor for this. The Jumbotron display at a sports stadium. Let's really pull this metaphor apart to understand the physical reality of the brain.
Let's do it. Consider a massive LED screen composed of millions of individual light bulbs. A single light bulb on that screen has no inherent meaning.
It's just a point of light.
It's just a point of light. Furthermore, that single bulb does not belong to any single image. It might be illuminated to form the curve of a player's face in one frame, the white of a football in the next, and the red of a corporate logo a moment later.
And crucially, the system is robust against failure. If one bulb burns out, the image of the player's face is still perfectly recognizable. The information is not stored in the bulb. The information is the pattern itself.
The brain operates on this exact principle. Donald Hebb proposed the concept of the cell assembly, a subset of interconnected neurons that fire together to represent a specific memory or concept.
So any given neuron in the cortex might participate in thousands of different cell assemblies.
Right. And this computational strategy exponentially, almost infinitely, increases the storage capacity of the system. You're no longer limited by the number of individual cells, but by the combinatorial explosion of possible firing patterns.
But this immense capacity introduces a terrifying mechanical vulnerability. And this is where the system's perspective becomes essential for understanding psychiatric illness. A jumbotron can display an infinite variety of images, but it is bound by an absolute physical constraint.
It can only display one distinct image at a time.
Exactly. This is the core problem of ensemble coding. The brain must actively prevent what systems neuroscientists call a catastrophic merge.
Let's conceptualize two separate cell assemblies to explain this. Assembly A represents the concept of a dog. Assembly B represents the auditory concept of a bell. Because of the nature of ensemble coding, Assembly A and Assembly B likely share a significant number of the exact same neurons.
Right, just like the jumbotron uses the same bulbs for different pictures.
If Assembly A and Assembly B activate at the exact same millisecond, if those two patterns attempt to occupy the network simultaneously, the shared neurons fire, the distinct boundaries of the patterns overlap, and the system physically merges them into a completely novel, unrecognizable cell assembly.
It creates Assembly C. But Assembly C doesn't mean dog and bell. It's just meaningless, jumbled static. It's a total loss of information. I always think of this like double exposing a frame of analog film.
That's a good way to put it.
Right. If you take a picture of a mountain, and then without advancing the film, you take a picture of a face, you don't get a clear picture of a face on a mountain. The overlapping exposure destroys the structural integrity of both images.
That is a highly accurate representation of the biophysics. The temporal overlap destroys the information. Therefore, a neural system utilizing ensemble coding can only function if it possesses a rigidly enforced, metabolically expensive infrastructure to coordinate timing.
The metronome.
The brain requires a relentless metronome to ensure that the cells comprising the dog assembly fire in perfect simultaneous harmony, but that the dog assembly and the bell assembly are kept strictly segregated in time, even if they are only separated by tens of milliseconds.
So timing is literally the ultimate bandwidth limit of human consciousness. If the metronome falters, our thoughts double expose, which naturally leads us to the physical infrastructure of that coordination. We always hear pop science references to brain waves, right? Theta waves for relaxation, gamma waves for focus.
Yeah, you see it on meditation apps all the time.
Exactly. But the text gets incredibly granular about the biophysics here. Where do these waves actually come from?
Well, we have to dispel the notion that brain waves are ethereal, macroscopic fields floating through the cerebral cortex. What we measure on an EEG, or more precisely with intracranial electrode as local field potentials, or LFPs, they are the summed biophysical consequences of millions of microscopic ionic currents.
Let's look at the cellular level. The text describes sinks and sources.
A neuron maintains an electrical resting potential by actively pumping ions across its semi-permeable membrane. When a synapse activates the cell, specific ion channels open. Positively charged ions, predominantly sodium or calcium, rush from the extracellular fluid into the interior of the neuron.
Because the ions left the extracellular space, that space suddenly has a deficit of positive charge.
Yes. That localized deficit of extracellular positive charge is what we call an electrical sink. And our recording electrodes register that sink as a downward negative deflection on the monitor. However, physics demands equilibrium. To balance that sudden influx of charge, passive electrical current flows out of the neuron at distant points along its membrane, returning to the extracellular space. This outward flow is called a source.
So an LFP, a brainwave, is just the synchronized sloshing of these ions in and out of millions of cell membranes, creating alternating sinks and sources.
Basically, yes.
But here's the mechanism that completely overturned my basic understanding of neuroscience. The actual communication between neurons, the fast spiking action potentials, are not what generate these massive rhythmic brainwaves.
It's a really common misconception. An action potential is a highly localized, incredibly brief event, typically lasting less than 1 millisecond. Because it's so fast, the electrical currents cancel each other out before they can summate across large populations of cells.
So spikes don't equal waves.
Exactly. Spiking activity contributes very little to the low-frequency oscillations, anything below 100 hertz, that dominate an EEG recording. The massive rhythmic swells that structure our consciousness are driven primarily by the slower, longer-lasting, highly synchronized currents generated by inhibitory neurons.
I really need to stop and emphasize this because it is entirely counterintuitive.
Yeah.
We are taught that excitatory neurons, the ones that release glutamate, turn the brain on, and inhibitory neurons, releasing GABA, turn the brain off.
Right.
How does a system designed to turn things off end up generating the synchronized rhythm of consciousness?
Because inhibition in a complex network does much more than just silence activity. Inhibition is the mechanism of synchronization. Let's return to the orchestra for a second. The excitatory pyramidal cells are the musicians. The inhibitory interneurons are the conductor.
But the conductor doesn't actually produce any sound.
Exactly. The conductor's primary job is to tell the musicians exactly when to stop playing. And by defining the silence, the conductor dictates the exact, precise moment when the musicians are allowed to play.
Ah, I see.
In the cortex, dense networks of inhibitory interneurons, specifically those expressing a calcium-binding protein called parvalbumin, fire together. They release massive rhythmic waves of the inhibitory neurotransmitter GABA onto the surrounding excitatory cells.
So they basically blanket the local network in a suppressive chemical fog. No one is allowed to fire.
But that GABAergic inhibition decays quickly. As the inhibition lifts, it creates a sudden, fleeting window of opportunity where the excitatory cells are free from suppression. If an excitatory cell has been receiving input and is primed to fire, it cannot fire during the suppression. It is forced to wait.
So when the window finally opens, all the primed excitatory cells fire their action potentials at the exact same millisecond.
The inhibition forces them to synchronize. It's like a dam holding back water. When the floodgates open, everything rushes through at once. And then, a millisecond later, the parvalbumin interneurons fire again, slamming the window shut.
Stop, go, stop, go. That rhythmic opening and closing of windows of opportunity is the primary generator of the gamma oscillation.
Yes. Gamma is a fast local rhythm. typically oscillating between 30 and 100 times per second, generated locally by this precise feedback loop between excitatory pyramidal cells and fast-spiking parvalbumin interneurons.
So gamma is the rhythm of local computation. It's what binds the individual cells into a cohesive ensemble code. But gamma doesn't travel well, does it? For long-distance communication, the brain relies on a different rhythm, the theta wave.
Theta is a much slower rhythm, oscillating between 5 and 12 hertz. While gamma is generated locally in patches of cortex, theta is a global sweeping rhythm.
Where does it come from?
In the hippocampal formation, it's heavily driven by rhythmic pacemaker cells located deep in the basal forebrain, specifically the medial septum and the diagonal band of Broca. Theta is the overarching rhythm of active sensory exploration, memory encoding, and REM sleep.
So we have the local fast rhythm, gamma, and the global slow rhythm, theta. But how are these inhibitory conductors wired into the network to create these dynamics? The text details 3 canonical pattern-generating networks, or motifs, that seem to be universally deployed across the brain.
Yeah, these are crucial.
Let's examine these microarchitectures. First up is feed-forward inhibition.
Okay, imagine a signal originating from a distant brain region, arriving at a local cortical column. The signal travels down an axon and excites a principal target cell. But that same incoming axon also splits off and simultaneously excites a local inhibitory interneuron.
Okay, so both get hit at the same time.
Because the interneuron is smaller and faster, it fires immediately, releasing GABA onto that same principal target cell just a fraction of a millisecond after the principal cell fires its initial spike.
It's a built-in shut-off valve. The incoming signal triggers the target, but also immediately triggers the mechanism to silence the target.
The function of feed-forward inhibition is to enforce extreme temporal precision. It prevents the principal cell from firing multiple sloppy spikes in response to a single input. It creates a razor-sharp, ultra-brief window for the signal to be registered, ensuring the timing of the ensemble remains crisp.
The second motif is feedback inhibition.
This is a classic self-regulating loop. A principal excitatory cell fires an action potential. The primary axon sends the signal downstream, but an axon collateral loops back to excite a local inhibitory interneuron. That interneuron then reaches back and inhibits the very principal cell that just excited it.
Like a thermostat turning off the heater once the room gets warm.
Precisely. It is a homeostatic mechanism designed to prevent runaway excitation. Without robust feedback inhibition, the excitatory cascade would amplify continuously, eventually resulting in a generalized seizure.
And the third motif, which seems absolutely vital for preventing that catastrophic merge we discussed earlier, is lateral inhibition.
Lateral inhibition is the architectural basis of decision-making and sensory discrimination. It is a winner-take-all mechanism.
How does that work in practice?
Imagine 2 competing cell assemblies. Let's say you were looking at an ambiguous optical illusion, like the classic Rubin vase, where you can see either 2 faces in profile or a single vase in the center. Both assemblies are receiving partial input.
But you can only perceive one at a time. The image flips back and forth.
Right. The assembly that receives a slightly stronger initial input will fire its action potentials first. As it fires, it strongly excites a network of interneurons. These interneurons do not inhibit the active assembly. They project their axons laterally, blanketing the competing weaker assembly in heavy inhibition.
Oh, so it actively suppresses the competition.
It exaggerates the tiny initial difference in input, allowing the slightly stronger thought to dominate the network entirely. It forces the system to make a definitive choice, preventing the visual cortex from merging the vase and the faces into a blurry, incomprehensible mess.
That's incredible. So we have these intricate micromotifs controlling local timing, but human cognition requires integrating information across massive spatial distances. How does the visual cortex, processing the sight of a dog, coordinate with the auditory cortex, processing the sound of a bark, and the hippocampus, accessing the memory of a childhood pet?
This is where the different rhythms have to interact. The text describes several forms of synchrony. Amplitude synchrony, where the power of 2 waves rises together. Phase synchrony, where the peaks and troughs align perfectly. But the mechanism that truly binds the brain together is phase-amplitude coupling, or PAC.
PAC. The text uses the term nesting to describe it, which I find highly evocative.
It's a great visual. In PAC, the phase of the slow global wave, the theta wave, actually modulates the amplitude or the power of the fast local wave, the gamma wave.
This was the concept I struggled with until I visualized it like the ocean. Imagine you are watching the open sea. You have massive, slow-rolling swells of water moving across the ocean surface. That is the 8 Hz theta wave generated by the deep pacemakers. Now, riding on top of the very crest of that massive swell is a brief cluster of fast, choppy, turbulent ripples. That is the 60 Hz gamma burst.
That analogy maps perfectly onto the electrophysiological data. If you look at an LFP recording demonstrating PSYCH, you do not see continuous gamma activity. The fast gamma oscillations, those intense periods of local computation where the windows of opportunity are opening and closing, they only occur at a highly specific phase of the slower theta cycle.
Almost always nestled right at the peak of the wave.
Yes. During the trough of the theta wave, the gamma activity goes completely silent.
Why is this nesting architecture so crucial for cognition?
It solves the integration problem. Gamma is fast, but it is metabolically demanding and spatially restricted. It can only synchronize cells within a few hundred microns of each other. It binds local features. It binds the texture of the dog's fur to the color of the dog's fur.
But theta is a low-frequency wave that can travel across entire hemispheres with minimal signal degradation.
Exactly. By nesting the local gamma bursts within the precise phase of the global theta wave, the brain establishes a universal clock. The visual cortex, the auditory cortex, and the hippocampus all experience the peak of the theta wave at the exact same millisecond. Therefore, their local gamma bursts fire simultaneously, allowing vast distributed networks to share data and bind disparate sensory features into a single, unified conscious moment.
Theta is the global delivery system, and gamma is the local packaging. They braid the microscopic and the macroscopic together.
Which brings us to the most clinically profound realization in modern psychiatry. If this intricate, nested temporal infrastructure is what constructs a coherent reality, what happens to a human being when that infrastructure cracks?
This leads us into the clinical manifestations, and specifically the discoordination hypothesis of schizophrenia.
For the better part of a century, biological psychiatry searched for the lesion. We looked for the damaged lobe, the absent neurochemical, the broken anatomical part that caused schizophrenia.
And we didn't find it.
We didn't. The discoordination hypothesis fundamentally changes the search parameters. It proposes that the hallmark symptoms of schizophrenia, the fragmented speech, the auditory hallucinations, the breakdown of working memory, the failure of reality monitoring, they do not arise from a structurally broken brain region.
The constituent cells may be perfectly healthy.
The pathology is a failure of phase-amplitude coupling. It is a loss of temporal harmony. The network is discoordinated.
The musicians are skilled, but the conductor has dropped the baton. And the evidence supporting this isn't just theoretical. The chapter details a landmark study using the psychotomimetic drug PSYCH on the spatial navigation networks of rats.
Yes, the analysis of this study is a masterclass in why systems-level thinking is necessary.
Phencyclidine is critical here because, in healthy humans, it induces an acute psychological state that is virtually indistinguishable from an acute schizophrenic psychotic break. It causes profound sensory distortions, paranoia, cognitive fragmentation.
In this study, researchers implanted high-density electrode arrays into the hippocampus of rats and recorded the activity of dozens of individual place cells simultaneously while the rat navigated a familiar circular environment.
They mapped the baseline. They knew exactly which corner of the arena triggered cell 1 and which corner triggered cell 2. Then they administered systemic PCP to the rat. What happened to the neurons?
This is the pivotal finding. If researchers analyzed the data using the classical single-cell dedicated coding framework, the results were baffling.
Why?
Because looking at any individual place cell in isolation, the drug had no apparent effect. The spatial tuning curves remained perfectly intact. Cell 1 still fired reliably when the rat walked into the north corner. Cell 2 still fired in the south corner. If you were a neuroscientist examining a single cell, you would be forced to conclude that the brain was functioning normally.
The individual violinists were still playing the correct notes when instructed. So where is the psychosis?
The psychosis only becomes visible when you abandon the individual cell and look at the ensemble correlation matrix. The researchers stopped asking where the individual cells fired and started asking when the cells fired in relation to each other. They looked at the temporal cross-correlations across the entire recorded network.
And that's when the reality of the drug effect emerged.
The entire correlation matrix was shattered. Even though cell 1 and cell 2 maintained their spatial preferences, the intricate, rhythmic sequence of how they fired together as a cohesive cell assembly was utterly disrupted.
The temporal relationships between the neurons had shifted dramatically.
The previously stable, coordinated ensemble code representing the familiar environment was destroyed, replaced by a completely aberrant, novel pattern of rhythmic firing.
But the text emphasizes a deeply unsettling detail about this aberrant pattern. It wasn't just random chaotic noise. The network fell into an aberrant stable state. And the system demonstrated multi-stability.
Multi-stability is a concept from dynamic systems theory. It means a complex system can exist in more than one stable configuration. As the PCP began to metabolize and wash out of the rat system over 30 minutes, the neural activity did not slowly and linearly transition back to baseline.
It snapped back and forth.
Instead, the network toggled. It would abruptly snap back and forth between the normal pre-injection network state and the aberrant PCP-induced network state.
Like a light switch flickering between two circuits. But the most profound part is what they observed before they even administered the drug.
Yes. Upon incredibly rigorous mathematical analysis of the baseline recordings, they found that even in the normal, healthy, unmedicated brain, the network would occasionally spontaneously flicker into that aberrant temporal state for a few brief milliseconds.
Wait, so the psychotic network state already exists within the healthy brain?
It is an inherent potential configuration of the architecture. What the PCP did, and by extension, what the pathophysiology of schizophrenia likely does, is simply change the thermodynamic probabilities of the system. It deepens the attractor basin for the aberrant state, causing the network to fall into it and become trapped, preventing the brain from resting stably in the functional, coordinated reality state.
That fundamentally changes how we view a psychotic break. It isn't an external invasion, it's the brain getting locked into an alternate rhythmic rhythm. But obviously, schizophrenia is rarely caused by PCP ingestion. The chapter makes it clear that the root of this discoordination is neurodevelopmental.
It is.
Let's look at the two prominent neurodevelopmental animal models discussed in the text that attempt to trace how this temporal infrastructure goes wrong from the beginning. First is the MAME-17 model.
Right. MAM, or methylxoxine methanol acetate, is a potent mitotoxin. It is a chemical agent that actively disrupts and halts cell division. In this established translational model, researchers administer a specific dose of MAM to pregnant rats on exactly embryonic day 17.
Why that specific 24-hour window?
Timing in neurodevelopment is everything. Embryonic day 17 in the rat corresponds to a massive, highly specific wave of neurogenesis and cellular migration in the developing limbic system, particularly the hippocampus and the prefrontal cortex. By halting cell division on that precise day, the researchers permanently alter the architectural trajectory of those regions.
And when those rat pups are born and grow into adults, what does the resulting phenotype look like?
They exhibit an array of physiological and behavioral anomalies that strikingly parallel schizophrenia. They display severe cognitive inflexibility on behavioral tasks. Their dopaminergic systems in the striatum are hyperresponsive to stress. But the most crucial finding from a systems perspective is a profound structural deficit.
What is it?
They have a massive permanent reduction in the number of parvalbumin-positive inhibitory interneurons in the prefrontal cortex and hippocampus.
The system develops without enough conductors to manage the orchestra.
Exactly. And because the interneurons are missing, the cross-scale coordination completely fails. Electrophysiological recordings show that the normal, precise temporal coupling between fast ripple oscillations in the hippocampus and sleep spindles in the prefrontal cortex is severely degraded. The anatomical pathway exists, but the temporal link is broken.
So a transient chemical insult occurring before the animal is even born permanently compromises the timing infrastructure of the adult brain.
Massively.
The second model the text outlines is the NVHL model, the neonatal ventral hippocampus lesion. And this model is fascinating because it explains the delayed onset of symptoms we see in human patients.
In the NVHL model, researchers induce a targeted excitotoxic lesion in the ventral hippocampus of rat pups on postnatal day 7. To translate that developmental timeline, rat postnatal day 7 is roughly equivalent to the third trimester of a human pregnancy. The injury is localized, and it occurs extremely early in development.
But the behavioral deficits, the hyperlocomotion, the cognitive impairment, the abnormal dopamine response, they don't manifest in the neonatal rat. They don't appear in the juvenile rat. The rat appears functionally normal until it reaches early adulthood. How does an injury sustained in infancy remain silent until adulthood?
This represents the essence of clinical systems reasoning. The physical lesion is in the hippocampus, but the ultimate functional failure occurs in the prefrontal cortex.
The executive hub?
The prefrontal cortex is the executive hub of the brain, responsible for cognitive control, working memory, and regulating subcortical dopamine release. Crucially, the prefrontal cortex is the absolute last brain region to fully mature, a process that continues through adolescence and into early adulthood, largely driven by the pruning of excitatory synapses and the maturation of those inhibitory parvalbumin networks.
And that maturation doesn't happen in a vacuum. The prefrontal cortex relies on incoming data.
Exactly. The developing prefrontal cortex requires continuous, highly structured, properly patterned rhythmic input from the hippocampus during adolescence to guide its final maturation. Because the hippocampus was lesioned on day 7, it spends the rat's entire adolescence feeding noisy, discoordinated, abnormal signals to the prefrontal cortex.
The prefrontal cortex is trying to calibrate its metronome, but the calibration signal is corrupted.
Consequently, the prefrontal networks fail to mature properly. Their inhibitory control systems remain functionally stunted. When the rat finally reaches adulthood, the prefrontal cortex comes online, but is essentially disinhibited and disorganized. It fails to regulate the mesolimbic dopamine system, leading to hyper-responsiveness.
And what I found so illuminating about this model is when the discoordination becomes apparent. If you take an adult NVHL rat and record its brain activity while it is simply resting in its home cage, grooming, or sleeping, the macro-level oscillatory power looks relatively normal. The system appears stable.
The structural deficits are masked during periods of low cognitive demand. The baseline rhythm is sufficient for basic survival behaviors.
But the illusion of health shatters when you ask the brain to do work.
Exactly. If you take that same adult NVHL rat and place it on the rotating active place avoidance arena, if you introduce environmental conflict, cognitive load, and the demand for high-level spatial calculation, the fragile network buckles under the pressure.
It just can't keep up.
The prefrontal and hippocampal circuits entirely fail to synchronize. The theta phase coherence between the two regions drops off a cliff. The rat cannot perform the task because the temporal infrastructure cannot sustain the computational load.
This maps so elegantly onto the clinical reality of severe psychiatric illness. We see patients who can appear perfectly lucid, capable of maintaining logical conversation while sitting quietly in an inpatient ward. But the moment they're discharged into the chaotic, conflicting, high-stress environment of modern society, or the moment they experience severe emotional distress, the network timing fractures.
Yeah, the orchestra can play a simple C major scale flawlessly, but the moment you hand them a complex Wagner symphony, the performance disintegrates into noise.
Because the underlying parvalbumin infrastructure, the inhibitory tone, simply lacks the robustness and plasticity required to handle the increased demand for temporal precision.
Spot on.
Which transitions us perfectly into integration and clinical reasoning. Let's pull the microscopic biology and the macroscopic clinical behavior together. If we summarize the core physical deficit driving this network discoordination, what is happening at the cellular level?
It consistently points back to the failure of GABAergic tone and parvalbumin expression. Post-mortem histological studies of brains from human patients with schizophrenia consistently reveal a significant reduction in the synthesis of GABA, the primary inhibitory neurotransmitter, and a marked decrease in the expression of parvalbumin within the interneurons of the prefrontal cortex.
And we know that parvalbumin is a calcium-binding protein. Its entire job is to quickly buffer and clear the massive influx of calcium that occurs every time the interneuron fires, allowing the cell to recover instantly and fire again at high frequencies.
If parvalbumin expression is reduced, that intracellular calcium clearance becomes sluggish and inefficient. The interneuron physically cannot maintain the high-frequency, rhythmic bursting required to generate precise gamma oscillations. The windows of opportunity it creates for the excitatory cells become wide, sloppy, and blurred together.
The metronome loses its crisp click and becomes a messy hum. And because the windows blur together, the network loses its capacity for lateral inhibition. It loses the ability to sharply segregate conflicting cell assemblies.
The thoughts merge. The distinction between an internal memory and an external perception degrades. The reality monitoring system, which relies on pristine phase-amplitude coupling to verify the source of sensory data, fundamentally fails, manifesting clinically as a hallucination or a delusion.
It is incredibly satisfying to trace that causal chain from a calcium-binding protein all the way up to a paranoid delusion. And a major reason this chapter exists is because we finally have the technology to observe this chain in action. The text highlights a few transformative leaps in how we record the missing middle.
The pace of innovation in neurotechnology is staggering right now. We are rapidly moving past the era of traditional low-resolution wire electrodes. The text specifically highlights NeuroPixels 2.0.
Which is just a masterpiece of silicon engineering.
It is a probe that weighs a mere 1.1 grams, thin enough to implant with minimal tissue damage, yet it contains over 5,000 densely packed recording sites multiplexed onto a single wire.
It allows researchers to record the individual electrical spikes of thousands of separate neurons simultaneously, distributed across multiple distinct brain regions, all while the animal is freely moving and performing complex tasks. They use complex spike sorting algorithms to isolate the individual voices of thousands of musicians in real time.
And we are not limited to electrical recording either. The text discusses the optical revolution, specifically the development of GCAMP8.
These are genetically encoded calcium indicators.
Yes. So using viral vectors, researchers can insert a gene into a specific subset of neurons that causes them to produce a fluorescent protein. This protein is designed to bind with calcium.
Right.
When the neuron fires an action potential, calcium floods the cell, the protein binds to it, and the entire cell physically flashes with bright green light under a 2-photon microscope. We can literally look through a cranial window into a living brain and watch the jumbotron light up in real time.
The implications of seeing the network in real time are profound for how we conceptualize treatment. Because if we accept that the disease is a dynamic, time-varying network discoordination, it forces us to admit that our current pharmacological tools are incredibly blunt instruments.
This is the ultimate clinical takeaway. Standard antipsychotic medications are static chemical agents. A drug like haloperidol binds to a D2 dopamine receptor and blocks it continuously, 24 hours a day, 7 days a week.
It doesn't know if the patient is sleeping, solving a math equation, or experiencing a hallucination.
It is static inhibition. It can blunt the overall hyper-responsiveness of the system, effectively lowering the volume on the static. But it cannot restore the delicate millisecond by millisecond phase amplitude coupling. You cannot fix a broken metronome by pouring wet cement over the gears.
The text argues that we need location-specific and time-specific correction, but we don't exactly have pills that know what time it is. So how do we achieve targeted temporal correction?
We use the brain's most powerful natural interface for inducing targeted plasticity.
Which is?
Lived experience.
Experience itself becomes the mechanism of action.
Precisely. The future of psychiatric treatment, as envisioned through this system's neuroscience framework, is the synergistic combination of biology and behavioral training. The goal is to use experience to induce targeted corrective neuroplasticity. We might use a targeted pharmacological agent or precisely calibrated non-invasive brain stimulation like transcranial magnetic stimulation to temporarily open a window of heightened neuroplasticity in the prefrontal networks.
You soften the clay. You make the rigid, aberrant network malleable.
Yes. And while that biological window of plasticity is open, you aggressively engage the patient in highly structured, rigorous computerized cognitive training, or specialized cognitive behavioral therapy. You force the patient to repeatedly execute tasks that heavily recruit the exact prefrontal circuits that are discoordinated.
The cognitive effort of the task physically drives the neural activity, and the artificially enhanced biological plasticity allows the network to physically rewire its synaptic weights and retune its oscillatory coordination.
You are not just masking symptoms. You are teaching the orchestra how to play in time again through rigorous, biologically augmented, supervised rehearsal. It is the ultimate synthesis of our understanding of the brain. The hardware and the software are inseparable.
That is an incredibly optimistic, proactive vision for the future of our field. It moves psychiatry from a paradigm of managing chronic decay to actively rebuilding the temporal architecture of the mind. Let's crystallize the massive amount of information we've covered today with our key takeaways. First, systems neuroscience demands that we look at networks of interacting regions. Brain function, and therefore dysfunction, arises from distributed circuits, not isolated areas.
Right.
Second, the core operational principles of the brain are integration, segregation, and hierarchical processing. The system must constantly balance the need to cleanly separate distinct thoughts with the need to integrate them into a cohesive narrative of reality.
Crucial point.
Third, cognitive and emotional processes depend entirely on the physical electrical timing of the brain, specifically phase and amplitude synchrony, beautifully illustrated by the nesting of phase-amplitude coupling. And finally, severe psychiatric disorders, notably schizophrenia, reflect disruptions in system-level dynamics. They represent a fundamental discoordination of the excitation-inhibition balance, a loss of the parvalbumin metronome, rather than a localized, broken piece of brain tissue.
That is a highly accurate synthesis of the chapter's core principles.
All right, let's pause and see if you, the listener, can put these mechanisms to the test. We are going to reinforce your learning with some single best answer style questions directly related to the systems we just unpacked. I'll read the scenario, give you a beat to formulate your answer, and then our expert will break down the clinical reasoning. Okay, question 1. If you are examining the microarchitecture of the cortex, what would you identify as the primary role of parvalbumin expressing GABAergic interneurons in the context of network coordination? Take a second to think. Okay, what's the answer?
The key mechanism here is that they generate fast gamma oscillations by creating synchronized windows of opportunity. Through high-frequency rhythmic bursts of inhibition, they act as the network's strict conductor. They force excitatory principal cells to hold their fire and then release their action potentials in tight temporal synchrony, which prevents the catastrophic merging of distinct cell assemblies.
Question 2. You are analyzing an LFP recording from a patient. You observe that the amplitude or the power of a fast 60 Hz signal reliably peaks exactly at the crest of an ongoing, slow 8 Hz signal. What specific system's neuroscience mechanism does this trace represent? Got your answer. Let's hear it.
The specific pattern represents phase-amplitude coupling, or PAY, often referred to visually as nesting. The phase of the slow global theta rhythm, the 8 Hz swell, is actively modulating the amplitude of the fast local gamma rhythm, the 60 Hz ripples. This is the brain's fundamental mechanism for cross-scale integration, allowing distant brain regions to synchronize their local processing into a unified cognitive moment.
Question 3. Consider the landmark experiment observing rat hippocampus place cells under the influence of the psychotomimetic drug PCP. Which of the following best describes the electrophysiological reality of the network after injection? Let's break this one down.
The profound finding was that the spatial tuning of the individual place cells remained completely intact. They still fired in their preferred physical locations, but the ensemble correlation matrix radically shifted. The network's temporal relationships toggled into a novel, aberrant, stable state. This illustrates that psychosis-inducing agents cause massive systems-level discoordination without necessarily breaking or altering the dedicated tuning curves of single cells.
Question 4. When discussing the canonical pattern-generating networks, which specific motif serves as an effective winner-take-all mechanism, selectively exaggerating differences in input to isolate a distinct cognitive representation? Think about the Rubin vase illusion. Okay, what's the verdict?
That mechanism is lateral inhibition. When a slightly dominant principal cell fires, it excites an interneuron that broadly projects and heavily suppresses the surrounding weakly activated principal cells. This sharpens the signal-to-noise ratio, ensures that only the most relevant cell assembly commands the network, and actively suppresses competing, conflicting representations.
Question 5. In the context of translational psychiatric research and the circle of being, what precisely does the phrase "the missing middle" refer to?
The missing middle refers to the vast, technologically challenging explanatory gap between microscopic molecular mechanisms, such as gene mutations or receptor binding affinities, and macroscopic readouts like clinical behavior or fMRI bold signals. Systems neuroscience attempts to bridge this void by explicitly studying the temporally organized action potential discharge within large ensembles of neurons.
And finally, question 6. In the neonatal ventral hippocampus lesion, or NVHL, rat model of schizophrenia, a structural lesion is introduced on postnatal day 7. Why do the primary cognitive and electrophysiological abnormalities, the network discoordination, only fully manifest when the animal reaches adulthood? Have a think about the developmental timeline. Let's hear the reasoning.
The delayed onset occurs because the core functional deficit lies in the delayed maturation of the prefrontal cortex. The early limbic lesion deprives the developing prefrontal cortex of the normally coordinated rhythmic input it absolutely requires during adolescence to mature its local parvalbumin networks. Consequently, the prefrontal networks fail to establish robust inhibitory control, a structural deficit that remains masked until adulthood when the animal is finally subjected to complex cognitive and environmental loads that demand high-level coordination.
Excellent. Be sure to return to the Kaplan and Sadok text to review the specific mathematics and histological figures supporting these models, because engaging directly with the primary data is the best way to solidify your clinical reasoning. Before we finish, I want to leave you with a final provocative thought to mull over. If our entire perception of reality, our ability to retrieve a cherished memory without corrupting it, our capacity to separate a hallucinatory whisper from a real voice in the room, if all of that relies entirely on the relentless millisecond by millisecond timing of inhibitory pulses acting as a biological metronome, how fragile is our baseline consciousness? Are our individual personalities, our quirks, our distinct ways of processing the world ultimately just unique, highly trained rhythmic signatures? If experience alters the network and the network alters the rhythm, does changing the rhythm fundamentally change the person? It is a profound reminder of the dynamic, ever-shifting nature of the psychiatric landscape we navigate every day. Here are the details of this episode.
Basic Systems Neuroscience. Byline. From parts to patterns, how distributed circuits give rise to thought, emotion, and behavior.